Fantastic. Well, welcome to the CNS talk, the first one of this academic year, and I'm very delighted to have Professor Aline Ferreira Correia, and my team is laughing now for me trying to roll my R's, from Wits University's Neuroscience Research Lab in Johannesburg, where she leads the division of clinical neuroscience. Aline is trained and licensed as a clinical psychologist and has devoted her work to studying rare movement disorders, especially Huntington's and Huntington's disease like 2, which we hear a lot more today. And she has also assessed more patients with HDL2 globally than anyone else. She's also a fellow of the MIND Institute, the Machine Intelligence and Neurodiscovery Institute at Wits University, and combines clinical work, neuropsychological assessment, and also brain imaging these days. And without further ado, I'm very excited to have you. Take it away.
Thank you, Stephanie. Thank you so much for the invitation and having me today. I'm not going to introduce myself more than that. Just going to tell you that from this point on, I'm going to call it HDL2 because it's just so much easier to do so, okay? And also just to keep in mind that I am coming from a perspective of a clinical psychologist or neuropsychologist, so I'm going to dive a little bit into genetics, but, it's not a genetist, so... Okay. So let's get it going, if this goes. Okay, let me try again. Okay, it's going. So today I'm going to be trying to convince you about how that talking about HDL2 is really important, and it's really also important to investigate. And after I convince you that we should all be thinking about HDL2, I'm going to provide a brief overview of what we know so far. So I'm going to start with some basic definitions and clarification of terms, because I have this fantasy that some of my HDL2 patients and students are going to access this at some point. So I'm hoping to make it accessible for people that are really, really unfamiliar with things that are probably very basic for you. So please bear with me. I'm also going to tell you a little bit about the epidemiology, describe the clinical presentation of this disease, discuss the neuropathology of it, and then tell you about the genetics and pathogenesis, which makes this like really interesting, an interesting disease from a scientific perspective, okay? And I'm hoping to close with what we want to know and what plans we have for the future as, researchers in the area of HDL2. Yeah, so that's about it. And within these topics, I am going to tell you some aspects that are really important to consider when I make really bold claims about what we know, which are some issues with research biases that we have in the investigation of HDL2, research challenges, and some of my personal and clinical and research experience. Okay. I don't know, Stephanie, if people are welcome to have questions as we go, or if we keep questions at the end, I'm fine either way.
That's really up to you. There is no strict rules, so whichever is fine for you works for us.
I'm fine with questions as we go, so we can keep the conversation going. Okay. So some preliminary thoughts. HDL2 is a neurodegenerative disease that is caused by a very, very rare genetic mutation. Okay, it's clinically and radiologically indistinguishable from another disease that is also rare called Huntington's disease, but we know way more about Huntington's. So for those who are unfamiliar with this term, a neurodegenerative disease is a condition that basically damages your brain in a gradual process, and it progresses until it gets really worse. So I decided, I thought, I don't know how many of you are clinicians, right? But I think for me to explain to you what I got into the world of Huntington's disease, it's because I started seeing patients with Huntington's disease. And I thought it would be important that you would see how this disease progresses or how this disease looks already in a somewhat advanced stage, so you can see the sense of urgency that I'm going to have when I talk about why it's so important to do this research, right? So I'm going to show you a very short video that it's uploaded on YouTube, just for you to see a clinical presentation of the disease, okay? So by looking at this video, I can I can imagine that you can have like a general characterization of Huntington's disease, but also Huntington's disease like 2, because as I mentioned, they are indistinguishable in terms of clinical presentations, right? So the reason why it's so important is because living with rare genetic diseases is profoundly difficult for the patient, most than anyone, but also for the family, okay? So there are many diagnostic challenges, right? I mean, HD it's quite known in the medical community, but HDL2 is not. So sometimes the diagnosis is quite delayed or it's completely absent, and it can be really difficult to access places where you can get the actual diagnosis of what is happening, right? What is explaining your symptoms. The treatment availability is quite limited or completely absent, which means that, sometimes you have to go through the experience of a rare genetic disease with no relief for your symptoms and no support in terms of medication and interventions. Also, the medical knowledge is limited, which is quite a sad part, because sometimes, and this happens often to my patients, where they need a specific professional, let's say a speech therapist or a dietitian, and they really don't know what Huntington's disease like 2 is and how it progresses and how to treat it at all. So, the medical community wants to help, but when there's not much information available, it's really difficult to find, to work with unfamiliar cases that you have no experience with. Also, there are limited resources in terms of places where you can go, where, here it's very difficult to access consultations with neurologists. For example, the African continent has the lowest ratio of neurologists per population, per number of people in countries, also psychiatrists. So even to make an appointment with a neurologist in a private sector can means that you have to wait for months and months. And in the public sector, it's even worse. And the same with genetic testing. It can be really difficult to access, and sometimes you have to wait for several months before you get the results. So it's quite a complex and difficult and triggering process. It gives a profound psychological burden for patients and families, particularly the increases feelings of alienation, isolation, guilt, fear, and uncertainty about the future. Which is it's a really difficult condition to live with and to progress in. Also, it's linked to major financial burdens because of the disability and the care needs that the patient needs, which means that people need to stop working before, really young, and stop providing to their families. And also there is no sufficient community support to kind of like hold patients and allow them to survive in a, dignified way if the family can support them. So, patients matter, and sometimes because this is so rare, people feel that, it's not that many people, so what's the point? But I'm here to tell you that, even if it's a small group of people, it's really important to support and to know about HDL2. But now living with HDL2 in South Africa is way, way more difficult than anywhere else in the world. I mean, the general picture of, healthcare in South Africa and in Africa in general makes it very difficult to access, to use, to identify, to kind of like live in a community of health support and make the survival of the disease for the patient and the family very challenging. Okay, but it's also important for science. And now I'm going to focus on this aspect because this is not a clinical presentation, but there are many aspects that HDL2 can enlighten us. And I think in general, HDL2 offers a unique opportunity to investigate the relationship between genetics and the brain, and how, our genetic makeup translates into a particular history for our brain and the progression of decline in our brains. Also help us to build models for common brain disorders, to understand brain neurodegeneration in terms of models and pathways, and not in terms of clinical presentations of disease. Also, it gives us an opportunity to explore therapeutic routes of treating, different kinds of movement disorders and symptoms. Of movement disorders and symptoms. So I think it's very important for science, and if scientists and funders kind of like put money on this, patients and families are going to benefit. Okay. So now going on the basic definitions and clarification terms before I move on with what we know about it. So when I'm talking about genotype, I'm going to be referring to genetic code, like all of the genetic characteristics of Huntington's disease like 2. And when I talk about phenotype, it's basically how those genetic characteristics express in something that we can see, like clinical presentation, brain imaging, and it's not only the consequences of our genotype, but also the consequences of how our genes interact with the environment, right? So we're gonna obviously know that even if we have a disease that is destined to progress, the progression is gonna be different based on our interactions with our environment. So that's, for us to keep in mind. So the basic definition of HDL2 is that it's an autosomal dominant disorder caused by a genetic mutation, okay? It's the exact same definition than Huntington's disease. And a genetic mutation, basically, it's a permanent change in your DNA sequence. It is autosomal dominant because normally we inherit genes from both our parents, right? One of each, we get 2 of them, and we only need to get a faulty one to definitely express the disease, okay? So if we inherit the mutation is going to be dominant because it's always going to be expressed. So we have a 50-50% chance to develop HD or HDL2 if one of our parents has it, okay? And it's autosomal because this mutation doesn't happen in any of the 6 chromosomes. Okay, so HDL2, it's also seen as a phenocopy of HD. So a phenocopy is when someone presents with the exact same clinical symptoms than a different genetic disease, but the cause is not genetic. Or the cause is genetic, but it's not the same mutation that the other gene is producing. So in this case, HDL2 mimics HD exactly, and that's why it's considered an HD phenocopy. Okay, there are several of them, and HDL2 is just one, and it's the one that is the most similar to HD. Okay. So how do we define a phenocopy for HD? It has to involve a movement disorder, so that's the core characteristic. It needs to have a negative genetic testing for HD. Obviously, that's obvious. If it tests positive for HD, it means that the movement disorder is linked to Huntington's disease. And it either has to have cognitive deterioration, psychiatric symptoms, or a familiar pattern of inheritance. And these 3 things are quite interesting to me because I'm going to tell you later on that perhaps HDL2 should not be characterized in the same way. Like, that this definition doesn't necessarily match what we are observing in HDL2, sorry. Okay. So now, in terms of epidemiology, when I tell you HDL2 is very rare, what I mean is that HD is considered a rare disease, right? And from when 100% of patients presenting with something that looks like HD get tested, 99% of them come positive for a mutation in HD. And only 1% comes with a negative test of HD. From that 1%, 97% goes undiagnosed. So they have a clinical presentation that looks like Huntington's. They have like a movement disorder, but it's not Huntington's, and no one knows what it is. Like there's not a test for it now. And only 3% of that 1% is an HD-like disease, like a phenocopy of HD. So from that 3% of that 1%, 30% of the patients are HDL2, have been diagnosed with HDL2. So it is really, really rare. And most of these patients are located in South Africa, which offers us as South African researchers and neuroscientists a great opportunity to do studies using methodologies that no one else in the world can use so far. A thing that I'm going to challenge in a bit. So in a review that we published quite recently, actually we published this review, I think the reference is there, last year. These are basically only 91 cases in the entire world have been reported. But there is a significant bias in this table, and sad to recognize that because I'm a co-author, but the problem with this table is that, for example, the Martinique, which is in the Caribbean, has a really large sample of HDL2 patients that I know. I have discussed with professionals there. They have seen these patients. They have been diagnosed with HDL2, but they have never been reported in the literature. So obviously, this kind of data is showing about two different kinds of biases. One of them, which I think is really, really interesting to think about, is that we only testing for HDL2 patients that look like HD. So if there is any atypical presentation of HDL2, most likely we're not picking it up. We don't know about it because we're not testing for it. But the second bias that we're having is that we're calling it a very rare genetic disease, but it may be rare because the people who are able to publish this data are not publishing it. It might be more frequent than we actually think. Obviously, there have been patients diagnosed in different countries that haven't made it to a literature. Okay. But you can see we have the largest proportion of patients reported so far. Okay. So now, in terms of clinical presentation, I mentioned to you before, and you saw in the video, and I'm sure you could see it easily with the patient that I show you, that HDL2 has, it's called a triad of symptoms, which is movement, psychiatric, and cognitive impairments, okay? They all progress in different ways, and this disease progresses to the point of developing a severe dementia. There is normally an aphasic dementia and an ataxic dementia that leads to an early death. Okay. So this is the point, and I put a disclaimer. I'm going to show you some of the videos that one of our PIs, David Anderson, did for his PhD. From the large study that we conducted. So we have permission to use these videos for educational research purposes, but we're not allowed to kind of like post them online permanently. So just to give you a heads up. Okay. So the first aspect that I'm going to show you is the movement disorder. It's one of the core clinical presentations of both HD and HDL2. And this is the kind of symptoms that you can observe in a patient, right? It's Correia, Parkinsonism, dystonia, rigidity, oculomotor alterations, speech impairment, and dysphagia. The major difference, even though they have exactly the same presentation as we can see here, is that for HD, this data has been collected from very large group studies. I think the larger involves like 30,000 patients from the study of Enroll HD, which involves a multi-center study from which South Africa was rejected to be part of, even though the neurologists that we work with put a bid to include South African patients. They were excluded, which is quite sad for many reasons, but one of them is because the genetic diversity of African patients is completely different from the rest of the world. Whereas for HDL2, we have very small, all of this data came from basically case studies or very small cross-sectional studies. And just to, kind of like clarify terms, Correia is an involuntary and unpredictable and jerky movements that constitute the core movement deficit in Huntington's and Huntington's disease like 2. Parkinsonism is basically an umbrella term that is used to characterize people that present with 4 core symptoms of Parkinson, but it's not Parkinson, which is resting tremor, bradykinesia, rigidity, and postural instability. Dystonia is a movement disorder that causes muscles to contract and then can produce motions of movements that cannot be controlled by the patient. And rigidity is increased muscle tone and the resistance to passive movements. And dysphagia is the difficulty swallowing. So you can see there is a very severe motor picture that basically impedes people from moving freely, from working. I mean, you saw in the video, the person can't even pick up his son. Okay, so we saw the movement symptoms, and now in terms of the psychiatric presentation, both HD and HDL2 present with depression, anxiety, apathy, and irritability. There's been always a contention of what is driven, right? If we have like a biological or, neuropathology that leads to this presentation, or whether this is circumstantial. Obviously, living with a neurodegenerative disease that causes so much disability can lead easily to depression, anxiety, apathy, and irritability. But, one would think it's both, right? OC symptoms like obsessive compulsive behavior have only been reported for HD and not for HDL2. And for HDL2, there are a lot of reports of personality changes. They are extremely vague. They much basically mean that there's something about people's behavior that changes, but they haven't been clearly assessed or operationalized in the literature, which is obviously one of the problems of naturalistic case reports in the sciences, right? Again, the reminder that, the methodologies are completely different, even though the picture looks identical. And in terms of cognition, both diseases present as a subcortical dementia with deficits in executive functioning, attention, processing speed. The deficits in memory have been inconsistently reported for both. There are definitely visoconstructive deficits. Problems with emotional recognition and working memory in HD have not been reported for HDL2. In the case of emotional recognition, it's because we haven't assessed that in HDL2. We don't have that kind of test in our battery, or no one has included in the battery before. And in working memory, because we do have that testing but the battery, but we haven't found these issues to be so predominant. So from this study, we published several papers, right? And the first one was a review, which I'm not talking about today, but we did a comparison between the HD and HDL2 phenotype, again, how they present clinically. And I told you before that they pretty much look identical, right? But something that we found is that it looks like HDL2 has a more severe clinical phenotype than HD. It's possible that HDL2 patients present 3 years earlier with more predominant dysarthria and dystonia. So this is the first group study, that allows for this kind of progression and discrepancies and control of variables that can now facilitate the identification of potential differences and dissociations between the diseases, right? However, when we send these videos to other neurologists that are experts in HD and movement disorders, they were blind to the diagnosis, they could not make a difference. So this is something we can only observe in group studies. In both disorders, the disease duration correlated with motor scores, which is something that we expect. As the disease progresses, the movement disorder gets worse, but this happened with all the motor symptoms except with Correia in HDL2. So then we published a paper on the neuropsychiatry of Huntington's disease, also a small group study that we didn't really observe significant differences between the neuropsychiatry of the disease, again, with one exception, which is that it looks like the psychiatric symptoms are quite more severe in HDL2 than they are in HD, okay? So motor symptoms normally are the reason that people start identifying the onset of disease in HD, but according to our assessment of HDL2 patients, the psychiatric symptom was the first to appear to identify the onset of the disease. So it is possible that HDL2 presents with a more predominant neuropsychiatric presentation instead of movement presentation, which makes, like a huge difference. And now, that part that I showed you, the definition of phenocopy needs to change. Okay. And obviously the neuropsychiatric symptoms and your capacity to have a functional life was dependent on the motor scores mostly. Then we published 2 studies on the neuropsychology of Huntington's. One of them was a series of control studies. Sadly, we don't have a group study here because each patient are at different stages of the disease, so they should not be grouped because we're characterizing different stages. Also, the group was too small. And what we did is that we used a methodology, the Crawford methodology, to compare one single patient with a well-matched sample of controls. And we compare here in South Africa, the variables that we selected is that people had to speak English as a second language, was an important issue. So we only select the controls that spoke English as a second language or as a third language, that attended a public schooling, because quality of education and whether school is private or public makes a difference in neuropsychological performance here in South Africa. And we also control for years of education and age as anyone would in anywhere else in the world, okay? And then we also did a comparison with Huntington's disease to see if we could identify any potential double dissociations in between these diseases, which means that, symptoms that are present in one disease, but definitely not in the other one. Okay. In terms of the neuropsychology of HDL2, there are obvious and consistent deficits in all of the patients, and they were mostly in the psychomotor speed and dexterity and visoconstructive domain. I mean, you would anticipate that given that this is a movement disorder. There were some deficits that were inconsistently observed in our patients that belong either in the executive functioning, memory, and learning and attention and concentration domains. So these were characterized as being moderately impaired in comparison to the visoconstructive and movements. Working memory tasks, which is a mystery to me, were mostly within the normal limits for all of the patients. Given that this is a disease that, affects the prefrontal lobes, it's interesting the working memory was the last to be affected or not affected at all. So something to think about. So the neuropsychological characterization of HDL2 is then a heterogeneous disease that impacts cognition in different ways. The cognitive symptoms appear early in the disease and evolves to dementia in a non-uniform manner, but it is in keeping with subcortical cortical loops of degeneration, particularly with the involving the basal ganglia. Okay. There were some differences that we could potentially interpret as a double dissociation between HDL2 and HD. We did match HDL2 patients to HD patients, but there are many disease-modifying factors that we can't match for, like age of onset, for example, plus controlling for all of the sociodemographic variables. So it's highly likely that these differences were due to like methodological artifacts. So I put them as like an interesting thing, but I don't trust these findings very much because of the methodology that we used. So now in neuropathology, which I mean that's, probably the most interesting part for the audience today. So what neuropathology do you think would lead to this kind of clinical presentation that involves movement, cognitive, and psychiatric symptoms? I imagine that all of you can guess it now. Basically involves the basal ganglia and the cortex. The most affected area in both of these diseases is the basal ganglia. I have created this graph because it really helps me to see which structures in the brain have been reported with the most atrophy, which are marked by the plus signs, and whether they are similar or not to HDL2. So, you can see like a comparison signs of like the 3 lines is very similar. The squiggly lines is somewhat similar. The difference symbol is, they are discrepant, and, which one presents with more atrophy than the others. So we have issues. The atrophy is mainly focused on the basal ganglia, particularly in the striatum and the globus pallidus, substantia nigra, and STN, also HDL2. There is cortical atrophy that is generalized to all of the lobes for both diseases, but mostly for HD. There are deficits in white matter tracts, but these have been more frequently reported for HD. And there are deficits in atrophy in the thalamus that looks to be more significant for HDL2 than for HD. So there's no reports of atrophy on the cerebellum or the hypothalamus or the hippocampus in HDL2 at all. So there seems to be, with the cases that we're having and the literature, that there is a distinction in the neuropathology of these diseases. So we published a study using a MRI brain. Lily, you have your hand raised. Let me go back to the slide.
Yeah, thank you. I was wondering here, when you say that there is no degeneration in cerebellum or hypothalamus, does it mean that there was study trying to find it, or is just not reported like for hippocampus?
So both. In one way, there are no studies that focus on these areas as areas of interest for HDL2, so we're not looking for anything in particular. The MRI reports are normally naturalistic, from MRIs that are taken from clinical files. So this is a neuroradiologist that is basically providing a description, and you can interpret that with whatever biases you may think the neuroradiologist can have in the clinical reports of MRIs. In our case, we were doing volumetric analysis and we did do it for the cerebellum, and we didn't find major discrepancies in between HCl2 and controls of the volumes on cerebellum and hypothalamus and hippocampus. So we did the measurements, but it was a very small group and there weren't significant differences with the controls.
But did you have in that study also patients with Huntington?
So we have, and I'm going to show you that right now.
Okay.
Yeah.
Okay. Sorry.
So we have, that's great that you are giving me that introduction, because we have a Huntington's group, a Huntington's disease-like 2 group, and a control group. And here you can see that for most of the areas that are highlighted in gray, the HCl2 and HD had more atrophy and controls, as you would expect, right, with a neurodegenerative disease, with the exception of the left and right thalamus, where HDL2 had more atrophy and HDs and controls. But if you see, for example, the clinical MRIs, the image 1 is an HD patient, and the image 2 is an HCl2, and the image 3 is a control from a sample, and the 1 and 2 are practically indistinguishable. So, we have to take the MRI data from the clinical studies and the single cases really carefully, because it's just like clinical descriptions of brains, basically. So, yeah. I don't know if that answers your question, Lilith. Is that?
Yeah, thank you.
So what that means is that there's a lot of potential for research there, which I'm hoping is going to, the point that I'm trying to sell here. Okay, so now in terms of the genetics and pathogenesis, okay, so HL2 again is a rare autosomal dominant genetic disease caused by a mutation. I already mentioned the mutation. It's a change in the genetic material that is permanent. They involve something that is called an insertion, which basically increases, this is going to sound a little bit complex, but increases the nucleotides in the genes by adding one or more. And I'm going to show you a picture that is going to clarify this. But basically, all the genes are formed by nucleotides. There are long polymers of molecules, right? And what happens is that there is an increase of particular nucleotides for each one of the disease that makes it go from normal, as you see at the top, like in the red area, from very, very long. And when they're very, very long, it basically starts creating a lot of noise. It can't perform the function, and it ends up breaking and creating like a toxic waste inside the neuron, if I can put it one way. So the mutation of HCl2 happens in a very interesting place, which is the GPH3 gene. Okay, this is a junctophyllin. There are several of them that codes protein for the neuron, right? It's brain specific, and it takes, the function is basically linked to the membrane complex of the neuron. So it has, and you can see here in this drawing, it provides structure, almost like a column that allows for the separation of the calcium channels. It allows for the structure of the membrane to be protected, and therefore is linked to the process of action, potential, and communication of neurons. So it has a function that is 100% linked to the neurons and the brain, okay? So it links also a plasma membrane with endoplasmatic and sarcoplasmatic reticulum in excitable cells. So this is like a simplified drawing. Allows for a crosstalk, and it looks like the presence of GPH3 has a significant role in neurons that have been attributed to functions of memory and motor coordination. So, as a general interesting finding, we know this because of the studies with rats. And this is a very fancy drawing that I'm just putting to show off that we had published in our review. And you can see a little GPH3 here in the right corner, linked to the endoplasmatic reticulum and providing structure for the neural membrane, which is something that I just wanted to show you in the big picture. And this basically is trying to describe the pathogenesis of HDL2 and how it damages the neuron. Huntington disease, on the other side, is caused by a mutation in a gene that is called HTT, or Huntington's, okay? The interesting aspect is the HTT is present in the entire body, not just in the brain, like JPH3. And it's linked to process of neurodevelopment and neurogenesis. So, the development of the brain and basically the creation and development of neurons. So it's everywhere in the body. So it's interesting that even though it's everywhere in the body, it presents as a neurodegenerative disease. So to summarize it, and because I have the brain of a psychologist, and I think with these little diagrams, basically a difference between HD and HDL2 is pretty much genetics at this point, right? So they both create mutations, are caused by mutations in different genes. HTT leads to HD, GPH3 leads to HDL2. HTT and GPH3 have different functions. HTT has the function of genetic transcription and intracellular transport, whereas GPH3 also has some functions in genetic transcription, but it's very much linked to cell excitation and structure in the neuron, right, in the membranes. So if they have different functions in the gene, right, when there is a mutation, there is a loss of function, right? That gene is not able to provide the same function, and that obviously leads to loss of the normal function, right? And after loss of the normal function, it creates an abnormal metabolism because they're not carrying their jobs and their weight, which reduces the resilience of the neurons, damages the homeostatic process in the brain, and leads to neuronal death. So can perform loss of normal function. However, the neuron also dies by all the pathological processes that are caused by the mutation, right? Which are aggregations. When those sequences of nucleotides break, it creates like noise and garbage inside the neuron. So there are called conformations and aggregations of proteins that interrupt processes and create a pathology within the neuron. They also have produce more mutant HTT protein, so it aggravates the process. And for HTT, for Huntington's, we know there is a process of excitotoxicity. All of this also leads to neuronal death, right? And in general, this basically is responsible for the cause of pathogenesis in the brain. Interestingly, and that's the reason why I have one thin arrow and one thick arrow, is that it looks like the pathology or the pathogenesis in HDL2 is more due to a loss of function, whereas for HTT and Huntington's is more due to a pathological gain of, damage in the neuron. So, we think that there are different kinds of routes to create pathology in the brain. Okay, so they are definitely then genetically different, that we know. They are not similar. Different chromosomes, different associated genes, different kinds of expansion. How big those expansions need to be are, quite similar, but we haven't got for example, such big repeats. Repeats is how long that expansion of the nucleotides are, and the longer they are, the earlier the disease is going to present. So, for example, in Huntington's disease, if the expansion is really, really long, you can have juvenile cases, and we don't have yet juvenile cases reported for HDL2. Somatic expansion, this is something that, it's a really complex genetic term, but basically it's the changes of the mutation that are not passed down from the affected parents. It changes in the actual body of the person that is carrying the mutation. There is a loss in HD of axonal transport and in HDL2 of calcium signaling. And anticipation is a process where every time a new generation in the same lineage is born with a mutation, the expansion grows, which means that the disease starts presenting younger and younger and younger. So in the HD2 case, if you inherit the mutation from your dad, you have a higher risk of presenting with the symptoms much younger than your dad did. We haven't established that in HDL2 because we don't have this kind of large longitudinal studies that cover several generations. Okay. So if you want to know more about the genetics, and this is a nice review that we published, that it has a very heavy genetic component. In case this is something that you enjoy, I invite you to download it and read it. So what are the plans with what we know, what we want to know now for HDL2, and what's my hope for research in the future, right? I put a unicorn because that's how it feels to look for patients with HDL2 here in South Africa and in the world. It's like this, really difficult to access sample, but we would like to, have the possibility of characterizing a completely unique genotype. There are many genetic studies that can be conducted in HDL2. There's also a unique pathogenesis. They are remarkably similar, but their path do diverge, and we're starting to identify with the first group study potential differences, and we think if we do much more large studies with groups, we can then now have distinctive characterizations of how the pathogenic this takes place. This is a lot of microbiology. We try that route. It's really hard because the donation of brains is something that is, complex to harvest, to maintain, to transport, but also in terms of cultural beliefs and whether you are allowed to donate your brain or not, depending on the culture you belong to. Definitely has a unique pathogenesis, so it should have anatomical differences, and we hope that those anatomical differences, we can pick them up with a better imaging, which is where our collaborations with Stephanie come in. We want to characterize better the phenotype in terms of neurological, neuropsychological, and psychiatric characterization. We can, we need to better assessments and better descriptions of our patients, and especially do follow-up studies. We want to know how the progression goes and whether actually HDL2 presents much worse than HD. But we need better methodologies, and we need cohort studies, and we need to follow up our patients from the not only onset, but pre-manifest and then onset and disease progression, which is, what we're hoping to in the future. But most of all, we're hoping that this is going to contribute to therapeutic approaches. That's really the end. The end is to help patients, to help the patients that I showed you at the beginning. I actually got the news today. It was published on the BBC that gene therapy worked improving HD, 75% improvement of symptoms with gene therapy in the UK. It's a research done by Tabrizi and Ed Wild. We have worked with Ed Wild in some of our publications, which are the leaders in the field of Huntington. So we are very excited because there is a possibility of delaying the presentation of symptoms that it almost looks like a cure, So we're hoping that this can also be the case for patients with HDL2. And so thank you. Thank you for listening. This is the research team. Dr. David Anderson is a neurologist. Amanda Krause is the medical genetics. Prof. Kay Cockcroft is one of the research psychologists and neuropsychologists. Marianne Gomez was our genetic counselor that we have to have in the team to help us in the process. And Dr. Mark Hagensen is the neuroradiologist that took the imaging and did the volumetric analysis for our imaging study. And these are our affiliations, the NHLS, Wits University, and the Wits University Donald Gordon Center, where we took our patients to be assessed. Thank you. Fernando.
Hey, thank you very much, Aline, for the presentation. It's very nice. Also, very nice diagrams towards the end. In the neurobiological side of Huntington's, and I was wondering, is it right to think of the disease as you were saying that the longer the sequence that patients had, the earlier the symptoms seem to arrive or seem to arise. So is it okay to think of it as a higher chance of aggregation of these proteins as they are doing their as they are being produced in the cell, which then leads to being accumulated, or why are the symptoms appearing earlier if the sequences are longer?
There is a loss of function and gain of toxic function much earlier due to the size of the aggregation. So it produces more mutant proteins, which therefore then now produce more aggregations and, early loss of function. So you 100% spot on that. Yeah.
Sweet. I think I have another
Lilith.
question, but I'm
There
gonna.
is something that I just want to add, which I find it to be really interesting because there is a potential of disease modifying factors in there, is that the identification of new patients can happen when you have a borderline size, right, that is not too big, and you start presenting symptoms maybe when you're in your 80s and 90s, so no one really knows that you had Huntington's and you die, and then the next generation there is a de novo case that presents later, perhaps when you're in your 50s and so forth. So, sometimes we think that it's really difficult to track the family history, but it's because exactly that aggregation makes the neurons die earlier and earlier, and then you have new mutations in your family line. Lilith.
Yes, thank you for the presentation. It was a really interesting topic, and I had no idea that there is Huntington disease like also a disease. Um, I have a question. So I did an internship in a clinic, and there was a patient with Huntington disease. And so this patient, we were doing like different neuropsychological tests just for every type of patients we were getting. And one of the tests that we did was on visual orientation task. And the patient really failed on that test. So you mentioned visual constructive processing are also damaged in
Yes.
patients. So later I went to and Google some papers, and it seems that the degeneration at the visual cortex starts earlier than motor. So I was wondering if that might be some different paths between HDL-like or HD diseases, that maybe the generation goes in a different way, because if you get the earlier, before starting the motor symptoms, you get visual degeneration, maybe in one type of disease, but not in another. So is there something that tells us maybe in the process something breaks earlier than in another one, or maybe one is more aggressively developing? So these are the questions that, because at the moment we have the snapshot, okay, they have the same like one of the final stages, they are similar, but maybe what that differentiates, yes, genetic, but also the how it comes to the final result.
So you're raising like two important issues there, right? One of them is the issue of assessment and how we can disaggregate the function to a point of being able to actually answer a question. For example, with typical neuropsychological batteries, it's really difficult to come to that kind of dissociation in between functions. So one of the things that we did is that we had some visual-constructive tasks that involve, actual construction with a pencil and, doing drawings, right? But we also had some visual-constructive tasks that will only require, like, let's say, mental rotation. But the problem is that with that mental rotation goes the issue of naming, for example, and the language. So it's very difficult to disaggregate those functions. So I do think that one of the steps that we need to take is exactly in that direction, in our capacity to do proper dissociation between functions and not just between texts, which requires a specific experimental task to, see where you're assessing. Because, for example, I use the Hooper Visual Organization test that I don't know if you guys are familiar, but it's a test where, it's like a picture is being broken like a puzzle. And you just meant to think about in your mind and rotate those pictures and put them together and think what they did. And one of the main obstacles for this test was actually nomination, like naming the picture. And even though what I decided to do is that they could answer in any language that they wanted, because sometimes it's because you can't find the word in English. There was an issue of nomination that perhaps could have gotten in the way of me to actually assess, visual spatial abilities in a way. So you have to be very precise in how you do that kind of dissociation. And the other issue that you're talking about, which is the issue of the actual, like anatomical degeneration, there's two comments there. One of them is that it looks like, there should be a specific pathological root for one, because GPH3 has specific functions and seem to be affected specific kind of neurons, at least in the mice models. So yes, I would think that perhaps the visual cortex gets affected before, if we think about it. But the other issue is that, for example, we know that mid-spiny neurons are the first one to go. And, there is a vulnerability of the mid-spiny neurons in the brain. And also the issue of, what kind of technology we use to assess this kind of pathological difference. So microbiology would be a best one, but it's really difficult. So now the next step is to try and see the brain with a better resolution, to see if we can see those kind of like changes, let's say in the visual cortex, before we see them in the motor cortex, when you take a more clear picture, which we don't have available yet. That's the plan for next year, hopefully. So it's a great question with a very complicated methodological answer that requires, like better tools of every, better MRIs, better analysis, better neuropsychological designs, etc. So yeah.
Yeah, because for example, concerning the first part, the visual constructive, we also did some tests that I think it's something look like it's for neglect mainly, or when you have some sort of a house that you have to just copy and then draw it again later on. Don't remember the name, but the person couldn't do it properly just because of the movements, because there you have to actually draw. So, but the test line orientation, you just have the lines and then a sample of the line and you ask which degree which one is it, like and the person was really failing this one, and I was just surprised because I thought it's not has to
That hard.
with primary visual like abilities. So yeah.
So you can clearly see that it's not movement, but movement does interfere though with a lot of the visoconstructive task. And movement also interferes with MRI, for example. Like we had some patients that could not do MRIs because, well, the whole thing about the process of doing MRIs on these patients, I can give a whole lecture, because from the first point, they're not familiar with the MRI. So I had to go there, hold their hands, do like a whole debriefing. But then it's also an issue of movement during the MRI. And, some patients are not suitable to be, to have MRIs. And then you can use sedation, but then that precludes a whole section of research that is, like task-based, let's say fMRI, that we won't necessarily be able to use with Huntington's patients in more advanced. So yeah, it gets very tricky to kind of like remove the movement component that is embedded in so many other functions and capacity to assess.
Yeah, even the study that you showed, the control one, the image was less noisy than patients with hands. So I was like, why is it? And then, oh yeah, but they cannot stay still, unfortunately. So yeah, thanks so much. Yeah.
Thanks. Muji, there was another hand up.
Hi, thank you for this is very interesting and informative. I just had a quick question about so you showed the gene that's affected in this disease. I forgot the name, J something.
PH3.
JPH3, and I was wondering if if this disease is only caused by a single gene mutation or multiple. So if and then are there any like transgenic or knockout mouse models, of this gene, and how do the phenotype, can be compared with humans? Like, do you have any information?
So yes, I love that question, because one of my absolute favorite papers ever is one paper by Stevanin, which has a description of a knockout mouse models with GPH3. So it is a single gene, and there are studies where they knock out that gene basically to kind of like remove the gene function, and the clinical presentation is pretty similar to the one that we have. It's got obviously a psychiatric presentation in mice is not provided, but there are problems with problem solving, with movements, so you can see that the clinical presentation is quite similar. So yes, we do have, and most of the HDL2 research that we can find that, is with knockout mice models. So yeah, it's fascinating, and there is equivalence with human behavior.
Yeah, okay. Thank you. That's very interesting. Thank you.
I love how you didn't need me at all to moderate the questions.
Sorry.
That's fine. It's very relaxing for a change. That's never happened before, I think. Great talk. There was a lot in there. I have a couple of questions myself as well. So one is that you nicely showed that there is cognitive differences between the two groups. And you made the distinction with memory, for example. And I was wondering if there is a specific neuropsychological cascade where they start with the same cognitive deficits and then follow a same trajectory, just in a shifted manner, or if it's completely different.
So I think, yes, there is a cascade, and I can potentially guess one based on the few cases they have like a full neuropsych assessment. And normally how I think it goes is that it goes with the visoconstructive and visomotor task first, right? Then it moves to executive functioning and then moves to memory. That's kind of like how I initially have conceptualized the progression. So executive functioning deficits are secondary to fine motor and visoconstructive deficits. And then learning and memory would be some of the last ones to be affected. That's kind of like what I'm seeing from the few cases that we have available.
Cool. And given all the similarities and differences that you presented, would you consider the two to be one disorder on the same spectrum, or should we actually classify them as distinct disorders?
So neuropsychologically, right, from a clinical neuropsychological point of view, I would see them as like one disorder. I think the discrepancies in neuropsychological presentation are going to be quite small. You could group them even, as one that can be distinctive for like other ones, for example, Parkinson's, dementia, or Alzheimer's type, because the cascading, the progression, and the clinical characterization is different, but I don't think it's going to be that different to make it a separate distinctive disorder. I don't think, however, it's going to be the same for example, how the onset and progression is. And the issue of progression it's an important one, because when you are working with patients, right, like knowing how fast the disease is going to take away their lives and the functional capacity is crucial for the therapeutic interventions and the family support. It introduces a relevant sense of urgency. So I do think that if it's something that is really important, if I could prioritize, it would be to investigate where this progresses much faster. I mean, that would mean that we really need to get going, and but I don't think it's going to be so different to move too far away from Huntington's.
Oh, thank you. I can see Fernando and Lila came back for round 2 of questions.
Yeah, I had a follow-up question on the symptoms. So the appearance of the symptoms is earlier, but is there also a continuum in the in the strength of these symptoms? Maybe some patients may have stronger symptoms than if they have the longer chain versus others.
So it depends. We think there are different kinds of motor subtypes, and in some cases, for example, the symptoms aggravate, for example, for the cases of Correia, but then they disappear because the rigidity gets, worse. So they don't all progress continuously, because some of them act in like an opposing ways. But they do have kind of like expected progression. We don't know with HDL2 because we think there are different motor subtypes. That's where we anticipate that they're going to be different types of motor presentation, and then they will have to be characterized differently. So yeah, we need to see how it goes with more research.
Yeah, I'm also wondering, this may be somehow a continuum within even healthy participants. It seems like this chain is I'm very fixated on the length of the chain, but I'm wondering if it's still present in healthy participants. There probably still may be some interactions that resemble Huntington's disease happening, but to a very much less extent, leading to healthy participants. I wonder if this chain has a known function.
No, it definitely has a function. So it's like, so I see what you mean. So we do have that because that basically forms our genes, like we all have it. And we all have it. Sometimes, I mean, I'm not a genetist, but there should be some kind of like inter-individual variability as there is with the brain, right? And perhaps different sizes, the sizes are acceptable until they become pathological. It's kind of like how tall you are, like you can have people of different heights, but it comes to a point where your height, can become a problem that doesn't allow you access, for example, to clothes, to shoes, to buildings, to elevators and things, which is kind of like how I make sense of these extensions. So I don't know if there are, what we know is that there is a range, and there is actually not agreement in the literature exactly at which point the pathological range starts, because there are some gray areas where we think there is potential for pathology, but it presents so late in life that it's really difficult to observe late in life. Like, perhaps this range only starts to produce symptoms when you're 110, like we know things at that age. But there is a gray area of, even in the literature, of when that size of the insertion starts becoming pathological. But there is a point where it's like a tip of effect, where this is bigger than this, 100% you'll develop the disease. So yeah, I hear what you mean, and I do wonder sometimes if certain kind of movement disorders that people present in the elderly years are linked to this kind of like genetic variations. I wouldn't know.
Thanks.
I can just
Lilith and
throw
then
in a
Valentin.
question from YouTube quickly. So Emra wrote a question to you on YouTube saying, thank you for the presentation. And then has a question for you from a more neurocognitive perspective, which is, we know that formulaic expressions rely on basal ganglia circuits, as known in various clinical populations. Given that HDL2 affects basal ganglia structures, have you observed reduced use or avoidance of formulaic expressions in patients' spontaneous speech?
It's so difficult to answer because there are already grammatical issues linked to speaking English as your fourth, fifth language, right? That are not pathological. We haven't done a good enough assessment of language for me to answer a question like truthfully. And I would imagine that any linguistic assessment will have to incorporate the grammatical nuisance of assessing a very diverse multilingual sample.
That's a very fair point, yeah.
We assess a lot of patients, but not enough to make like a generalization in terms of language. What is very observable is the issue of dysarthria. And I don't know, there are cultural issues also that kind of like intervene in this kind of exploration, because if you don't speak English well, right, but you're in a medical space where your assessor, in my case, it was actually a great thing that my English is not great, because it makes people feel at ease. But in some cases, if there is a significant discrepancy with the language, patients become less participative. For example, during the assessment, during spontaneous speech, they speak less to us. They also, if there is any kind of dysarthria, it becomes even more isolating. So it's very difficult to kind of like capture. You have to elicit it with a specific battery, which we haven't. So I can't even give you like a naturalistic assertion on that specific point.
That
But
makes sense.
it's amazing. That's one of the things that I would like to do, is to improve my language battery.
I'm just going to stop your video sharing now because you're cutting out, but don't let that stop you from taking more questions. Sorry for jumping in, Lilith.
No, it's okay. So I got another question. But for example, how I don't know, how do they diagnose Huntington disease, or is it just they do genetic testing, like this is the first thing? Because I was wondering how many of the Huntington disease studies actually are about only Huntington disease. Is it possible that those studies that we know about it, they also contain patients that were not Huntington disease, but Huntington disease like?
No, so the HD literature is quite developed, and you wouldn't have any kind of like mix up when it comes to a genotype that they're investigating, right? So they have already the families identified, tested, it's a single gene, and you can find it. So you can when it comes to these autosomal dominant diseases, you can take the patient at 2 points, right? One of them is the predictive testing, which is the person is not presenting with the symptoms, right? But there is some kind of risk because one of the parents got the mutation. So the person looks fine and they want to investigate the genetic status, which, whether they have the mutation or not. So they're asymptomatic. So that's a group. It's called like asymptomatic. Then you can be asymptomatic and be tested and have the mutation, but still not having the onset of symptoms, which is called pre-manifest. It's like I have the mutation, but I haven't yet shown the symptoms. But the genetic mutation is confirmed. And then you have the other group, which are the diagnostic patients, which they come to consultation already with the symptoms, and you do the testing to confirm the genetic status. And the testing is available, and you wouldn't move on. For example, in our study, before we invited patients, we were blind to the diagnosis, but the genetics knew exactly who had which mutation. They were all tested and confirmed mutations, and there's no risk of confusion because it's like a single gene and it's an easy mutation to investigate, and to define, basically.
Thank you. And I also found it interesting that HTT gene is all over the body. But are there testing of like some changes that maybe happen in whole body? So in organs, I don't know. Is it
Yes.
Hmm?
So Huntington's comes with some kind of like concomitant diseases. I mean, I haven't done the genetic kind of like literature review to know whether these are due to HTT mutation, but yes, having Huntington's increases your risk. Having Huntington's disease increases your risk for a whole array of concomitant issues. They go from, eye problems, but the problem is that we don't know if it's linked to the movement disorder, but there are gastrointestinal issues, there are eye issues, so yes, and I wouldn't know exactly whether it's driven by HTT itself or if it's like a cascade link to the other disorders. So I don't really know.
Okay, interesting. Thank you.
Valentina, I think you're next.
Thank you for the talk, Alina. I wanted to ask you a curiosity, actually. So there is evidence that Huntington's patients may not show awareness for the involuntary movements. And I was wondering whether this is the case also in the variant, especially because the variant shows, as you said during your talk, symptoms manifestation way earlier than Huntington's.
We didn't do like an awareness or an anosognosia like investigation in our patients, like a formal one, but I can tell you that that's most likely a case because some of our patients, for example, in the way we know our patients, we were in a clinic together, and it's really amazing how difficult it is to report their own symptoms in many ways, to kind of like characterize their own disease. And it's normally just the family member or the caretaker who is the one who reports that, especially with the cognitive ones. I was really impressed with the fact of, people with like advanced cognitive decline were reporting like a complete normal cognitive performance. We had one of our patients that was employed, right, was working, was the breadwinner of the house, and I was really excited because finally I can do like a full assessment, and when the patient sits in front of me, the patient with like an advanced dementia, like advanced. And I go to a neurologist and I'm like, this person has an advanced dementia. He said, like, how is this person still working? And it took me a while to figure out what this person's job was. Like, I was there for like half an hour trying to figure out what is it that is your job. And the job was cutting rope. Like he was some kind of manual worker in a line where all he had to do was like cut a rope that was passing in front of him, right? So, imagine that kind of unawareness of realizing, I don't even know how this person knew that he still had a job. That was how advanced the dementia was. So yes, I would say definitely it has to be an issue of awareness.
And
I don't know if it's exclusive motor symptoms, but yes.
no, and this is very interesting. And do you think it's the case where people who show higher levels of dementia, like most of dementia symptoms, are also the most unaware?
Yes. I think actually on awareness it has to be linked already with more advanced stages of the disease.
Okay.
I mean, I have picked up patients of my HCL2 patients from the side of the highway walking to like a doctor's appointment lost on the side of the road. And they have gotten into my car without knowing who I
Oh my God.
am. And I'm like, I'm your psychologist. Like, I know your name. This is your name. Where are you going? Like lost in the city. So yes, I would think that, and this is people that have like an advanced cognitive decline, but we're still mobile. The Correia wasn't advanced, so it does seem that there is something that precedes the motor decline in terms of cognitive awareness. Yeah. But this is an N equal 1 kind of experience. I don't want to make like a claim of characterizing the disease when I'm telling you our experience with a very limited number of patients. Yeah.
No, we should totally speak about that. No, and then I was wondering whether this is the case also for Huntington's.
Yes, it is the case for Huntington's.
Okay, so
I don't think the unawareness
it is
is
not
severe.
different between the two variants.
I
Okay.
don't think so, but I think it comes late in the condition. I also think there's like a weird overlap that for clinicians like me, it's really difficult to differentiate between unawareness and apathy.
Yeah.
Like I still wonder, like how would someone draw a line as a clinician of like how much is this, like severe apathy versus how this not kind of like connecting. For example, something that was really striking in regards to unawareness is the fact that Huntington's disease is known by the significant increase in suicidal ideation and suicidal attempts, right? It's one of the reasons why there is a particular predictive protocol and support structure for patients, and you would understand why someone with such a severe movement disorder would suicide. With our HDL2 sample, not a single patient, not a single one reported any kind of suicidal ideation. And then you wonder, is this cultural? Like, is this something about, culture that makes that as a no option? Is this complete lack of unawareness that doesn't even give existential dread that is linked to kind of like suicidal ideation? So we do have those kind of like Easter eggs hidden that points that this could be something important, but we don't really know what it is yet.
Okay. And do you have data questionnaires on their unawareness levels?
No particular in terms of unawareness.
Okay.
And I do think unawareness is better measured when it comes like experimentally, but yeah, no.
Okay, all right. Well, see you in Bordeaux, right?
Yes,
We can
yes.
continue talking about this in Bordeaux, I think.
Yes.
Right. There's one more question that came in on YouTube, which is starting with thank you for your presentation. And the question is, would the therapeutic approach be the same if the two mutations did not affect the same proteins? The treatments would be solely somatic in that case?
So now, because of the fact that there's actually a treatment for Huntington's, So far here in South Africa, we don't have genetic therapy for Huntington's yet, right? It just came out and it's just published. Normally, the treatment approach is very similar, and it normally involves like antipsychotics, like a lot of antipsychotics and psychiatric management and neurological follow-up. So the treatment is quite similar because it's just symptomatic, presenting very similarly. So yeah. So normally you end up medicating the all of the psychiatric presentations, the depression, the anxiety disturbances, the aggression, the core things like that. So yeah. And psychologically also we use the same approach as you would with a genetic neurodegenerative mutation. It's a similar approach.
Cool. Thank you so much. That was a very long session now. Sorry for keeping you so long on a public holiday for you. But thank you so much for joining us. Thanks everyone for being here, for engaging and asking your question.
Yes, thank you for your questions.
Have an exciting time, and I see you next month for the next CNS talk. Bye.
Bye.